will record the lectures and share the recording link to you all as well. But for the time being, just these will be notes for me. It can be shorthand. It does not have to be elaborate, grammatically correct sentences.
Yes.
The note that I need from you is examples of transcription factors. Question 1. Example of signaling pathway molecules. Question 2. Where they act as dominant alleles and the mechanism of action. Okay. Yes, Piyusha. Now you go ahead.
Sir, first I will like to ask about the binding site of an enzyme and what do we mean by the substrate, and also KD factor which you told earlier.
Okay, apologize. These are technical terms. I should have been careful. So, what is your background, Piyusha?
Sir, I did my BTech and MTech in electronics and communication, and my MTech specialization was biomedical signal processing.
Okay, fine. Take it. So now, what is a substrate? A substrate is any chemical. It can be another protein as well, or it can be DNA, it can be RNA, it can be a small molecule to which an enzyme binds and acts upon. Okay. Now, the enzyme, for it to convert a substrate to product, it must bind to the substrate. Okay. So, if you imagine for a minute that there is a solution, there are protein molecules that are enzymes, and there are chemicals that are substrates. Okay. Now in biological system, all bindings, like there are few exceptions, rare exceptions. Other than that, all bindings are reversible in nature. Okay. That means that the two components, they bind and then they dissociate. They bind and then they dissociate. So therefore, at a steady state, there will be some that are bound to the enzyme, like there are some enzyme molecules that are bound to the substrate and some that are not bound to the substrate, right? And this will be dictated by the affinity of the enzyme to the substrate. And how will the affinity be dictated? Typically by van der Waals forces and ionic interactions. You know, hydrophobic interaction, hydrogen bonding interaction, ionic interaction. Those will dictate how well the substrate binds to the enzyme. So, if the affinity is very high, then at a lower substrate concentration, you will be able to saturate the enzyme, because every enzyme, whatever piece of substrate it sees, it will bind and remain bound to it. Now this remain bound to it, I want to clarify that. Two enzymes having different affinities towards their relative substrates, okay? Now we are talking about two different systems. In one system, there is an enzyme with its own substrate. Another system, there is another enzyme with its own substrate. Let us imagine that the first enzyme is X and the second enzyme is Y. Let us imagine that X's affinity towards its substrate is more than Y's affinity towards its substrate. If you add the substrate to the same concentration, then you can imagine that at any given point of time at the steady state, more of X will be bound by its substrate than Y. Do you get it, Piyusha? Okay. Now, the way KD is defined is in a system like X plus its substrate, how many of X are in the bound form and how many are in the unbound form. That is KD is dissociation constant. Okay, so it is purely a reflection of the affinity of the enzyme to the substrate. Good. Next question.
Sir, I had a question.
Yes.
Sir, uh, what is wild type? Because everything found in nature is the result of
Sir, I
evolution, and
had
there is
another
some.
question. Actually, it was from the very introduction part.
Okay, I'll come back to you, Shora.
Okay, sir.
Piyusha, go ahead.
Sir, you told about codons. So
Uh,
actually, I also tried to look into Google, but did not understand much about the codons. And also, you were discussing about the codon bindings in the very first lecture.
uh-huh.
So if you can just explain a bit about the codon, and also if some source, if you can mention where I can just get some very basic idea about these things.
Okay, so explaining codon now is not appropriate because I have a full chapter on translation and translation regulation. I will come to it, but I will post in the group some easy to understand, source for codon, yeah, for you to understand codon. Okay, yeah, sure. Yeah.
Yes, sir. Sir, what is called the wild type? Because everything we found in nature is the result of evolution, and there is a
Yeah.
mutation in every genome. Though, what is called the wild type, and what is the difference between wild type and mutant?
Yeah, that is a $1,000,000 question, and I do not know the answer to it. Okay, I will tell you what is the rumor for human genes. The rumor is that uh, Craig Venter, who actually was successful in sequencing the whole genome, apparently it was his genome which was sequenced. Okay, and apparently those are the sequences that are referred to as the standard or RefSeq, reference sequence. Okay, but it is a really fundamental but difficult to conceive kind of a question, that if the gene or the allele varies from everyone, like right now there are 30 participants in this group, so there is certainly 60 alleles for every gene. Now which of these 60 will be the wild type? uh, it is mostly, it started as an operational definition that those who are behaving normal for any particular aspect, someone might have learning disability but can walk perfectly all right. So the walking function is normal. Okay, so those who are normal, their gene sequences were assumed to be wild type. I will give you another insight, which is perhaps you will find it startling, that when we talk about yeast genetics, we use a particular strain, W303 alpha or W303A, as wild type. But lately, there have been experiments done where Saccharomyces cerevisiae, the yeast, in the wild, okay, I made a distinction in my lecture about wild and wild type, right? In the wild, they can do certain things that our wild type yeast strain cannot. To elaborate it further, if wild type strain has, like W303 alpha, if it has 10,000 genes, we do not know function of more than 6,000 or so. Okay, while it is so easy to get to functional analysis in yeast. Okay, and the guess in the scientific community is that the genes for which we cannot define function under laboratory conditions, maybe they are important in the wild, in their natural environment. Okay, so yes, it is a little convoluted problem, and I think it will remain that way for foreseeable future. Please apologize. My apologies. I will have my breakfast while I am talking to you all. Okay, I woke up pretty late.
It's okay, sir. Sir, I have a question. Sir, so like whether we will always call wild type as dominant?
No.
No, because like if a mutation, like we know a natural wild type gene, now due to a mutation, it has become, you know, more functional, or more protein is being produced. So in that scenario, will the definition change?
Stop.
Yes, sir.
More protein is being produced generally does not come under the definition of allele.
Okay.
Alteration of the function of the gene product comes under the definition of allele.
Okay.
Okay, now continue.
Yeah, sir. So like if that is the thing, will we call like the mutated gene as dominant and the previous dominant gene as recessive now?
Absolutely.
Yeah.
Because
Okay.
in many types of cancers, certain classes of genes, like one that is very famous is RAS.
Yeah, yes.
This is mutated and it becomes more active than its normal counterpart. So it's a dominant mutation. No, wild type is not by default. I am talking about in a heterozygous where the other allele is recessive, then by definition the wild type allele is dominant.
Okay. Yes, sir. Clear.
Hello.
Sir, I have a question.
Go ahead, Saurabh. Sandharv, sorry, go ahead.
Yes, sir. Yes, sir. Sir, sorry, my video, I mean, whenever I'm allowing the video to open, it is getting disconnected. That's why I shut it down.
No problem. No problem.
Sir, I wanted to ask, yeah. So actually, I read it somewhere that most of the mutations occur in the recessive allele, like because mostly the mutation, mostly whenever the mutation occurs, the activity of protein gets decreases. So mostly the mutation occur in recessive allele and not in the dominant allele. So why does it happen? So how does the nature chooses the most of the mutation to occur in recessive allele and not in the dominant allele?
Okay, first of all, language. Okay, I am a stickler for language. So the mutation does not occur in the recessive allele or in the dominant allele. Mutation occurs in the wild type and makes it a recessive allele or a dominant allele. Okay? First. Second is I have not read the particular sentence that you were talking about, but I can completely rationalize why recessive mutations are more frequent. See, we are products of evolution. Okay? So few million years of experimentation has happened for us to have the enzymes that we have today that are functioning optimally. Okay, so majority of the cases, if you change an amino acid, chances are that the activity will go down. It is relatively rare to hit an amino acid and change it in a manner that the activity will go up. So that is the reason why you see recessive mutations more frequent than dominant mutations.
So, since the mutation is like now, so why will it happen that mutation will allow the protein to decrease activity most of the time, and not creating a protein
That's what I'm trying to say.
if it is random?
That through nature's own experimentation over last million years, there have always been mutations. Those that were not fit were discarded. So what we have today is perhaps the most optimal enzyme. Now,
Yeah, yes, sir.
if there was a possibility for it to be a better version, perhaps we would have that as the wild type today. So therefore, any change from what we have today is more probabilistic. Okay, it is not a rule. More probabilistic that it will lower the activity than it will enhance the activity. It's purely a matter of probability.
Yes, sir. Okay.
Sir,
And so,
I have
no question.
Okay, go ahead.
Yeah, okay. Sir, I wanted to ask one more question. I mean, if there's a mouse and it is having some black hair color or some brown hair color, and the gene which is responsible for the color of the mouse, for the color of the hair, having two alleles, like one responsible for brown and one is for suppose white. So now, if you switch off one of the alleles, you can switch off one of the alleles, right?
Mm-hmm.
We can switch off one of the alleles, like one of the allele which is responsible for white color. So if you switch off one of the alleles, and now the like the only which is remaining is black one, so it will show black color, right? Now if we switch off the other allele too, like the black and the white both, then what will the color of the mouse?
So there are few assumptions that you have made here. First, any coat color other than white is a function. Absence of that function makes coat color as white. Okay, number one. Number two, it is technically difficult to make allele specific mutation. I'm not saying impossible, but it is technically difficult. But what is easily done is a homozygous brown color can be met with a homozygous some other color, and it is typically the darker shade will prevail. Okay, so if you have a homozygous brown and homozygous black, the progeny will be black. It is because you think of it biochemically, the coat color is because of production of pigment, and how much pigment is produced and of what color will dictate the coat color. So if black is produced on brown, then obviously black will predominate, right? It is pure common sense or biochemistry, you can say. But Ubaid, you had a question.
Yeah, yes, sir.
Ubaid, you had a question. Please go ahead.
Sorry, sir.
Yes, sir. Sir, I have a question regarding phenotype genotype analysis. Uh, for analyzing the function of any particular gene, so you told that we mutate the one gene at a time, so that whatever the phenotype is that gene, we can over that function.
Yes.
In case if there are more than one gene or more than two genes that give the one phenotype, in that case, how we will discover that these are giving one, three genes are giving one phenotype? Do we have to mutate the two genes at the same time?
Yeah.
And what is the probability that two things we can combine?
Excellent question. A little bit of bragging. I have handled that kind of a problem myself. So if you look at my publication record, I published a paper in 2005, I think. That was precisely the purpose. So what happened was that the signaling pathway that I work with is called BMP signaling pathway. Okay. In flies, there is only one BMP called DPP. In vertebrates, we have 20 different BMPs. Okay. Now, it was known that BMP is likely to be critically important for bone formation. But when you knock it out, any of the BMPs, there is no bone formation defect. So what I did actually is I worked with 3 different BMPs, and I had reasons to work with those particular 3 and not all 20. I'm not getting into that right now. And then I knocked them out one at a time, and I found that there is no phenotype. And then I combined them. And only one particular combination gave me the phenotype, that is, there was no bone, 0 bone. Okay. Now, to impress upon you how critical your question is, that between 2 genes, BMP2 and BMP4, there are 4 alleles, right? 2 for BMP2 and 2 for BMP4. If I knock out up to 3, okay, just 1 allele is left, even that mouse behaved normally. Only when I knocked out the 4th one, there was this catastrophic defect of no bone. Okay, so yes, what you were saying is correct, that if multiple genes have redundant functions for a particular phenotype, then you will have to knock out many of those genes together to get any phenotype. Another classic frustrating example for vertebrate developmental biologists is the Hox cluster. So some of you may know that the Hox clusters, H-O-X, Hox clusters, they define our body plan. If you read any papers on Hox mutation, you will see that typically 3 to 4 genes are knocked out together before any phenotype is apparent.
Okay, sir. Sir, but if in any organism, if there are 10K genes, so can we come to conclusion that lot of genes are not discovered because maybe the 1 and the 10K gene have the they have the common phenotype, and they are scientists have not mutated all the genes in the probable combinations.
Mm-hmm. That is very much possible. So, when it comes to yeast, because today as the genome sequence is known, so you can predict the likely function of the gene. Okay. And then you can knock it out, and you still do not find any phenotype because there is a redundant function. There are many cases now coming up, which I referred to in my first or second lecture, that the way gene function was discovered are two diametrically opposite approaches. When geneticists did it, they started from a function and ended up in a gene. When biochemists did it, they had an assay which is in the test tube. Okay, and they tried to see that which particular protein or protein complex in the cell can execute a particular reaction. And they ended up finding the protein or protein complex. However, as you can imagine, that this whole investigation is being done in a test tube. So this will not tell you in any manner whether the same gene or the same protein or protein complex may have another function. Okay. Now, these kind of second functions are being discovered for many genes. I gave the particular example in either lecture 1 or lecture 2 of GAPDH. GAPDH is glyceraldehyde 3-phosphodehydrogenase, which is one of the most prominent enzymes in glucose metabolism. But today it is known that GAPDH can also act as a transcription factor. So unless we develop full comprehension about genes and its functions, it will be difficult to, like if a knockout experiment does not give you a phenotype, it will be very difficult to interpret those.
Sir, I have a couple of questions.
Sure.
So the assay you mentioned for the BMP, is it does it come under complementation analysis?
No. In mouse, it is very difficult to do complementation analysis. Okay, that is possible in yeast. We need to understand the merits and demerits of every model system. If I am trying to investigate the genes that are important for bone formation, then yeast or Drosophila or C. elegans, none of these are useful for me because they don't make bones. So I have to go to mouse, which does not offer many of the advantages of yeast or Drosophila or C. elegans, but allows me insight into things that are relevant. Next question.
So can I skip to yeast genetics since we're discussing about that? Yeah.
Okay.
So I have a question on tetrad dissection, the segregation of alleles experiment. So if I'm not wrong, you mentioned that in certain kinds of genes which are essential, the mutation can be directly performed in diploid. And if the genes are for other genes, they can be performed in the haploid, right? So my suggestion was, why we can't perform all those mutations in the diploid organism itself. Since if the mutation we are doing is going to be dominant, the diploid doesn't survive. Or if it's recessive, we can do tetrad dissection to see if it's separated in 2:2 ratio.
Very good question. So you name a function that it does, then I will give you the answer.
Okay. So
Name it.
galactose metabolism is finding.
Right?
Yeah.
So if you make the mutation in diploid, and as Sandharv discussed, majority of the mutations will be recessive in nature. So after mutation, when you plate them on a medium lacking galactose, what will happen?
Can't be able to survive.
Why? The mutations are recessive, right?
If it's recessive, it can survive, but if it's dominant, it can't survive.
Majority of the mutations will be recessive, as Sandharv pointed out.
Yeah.
So then what will happen? All will survive?
We can still expect the diploids to survive in the medium.
Yeah, you do not expect the diploids will survive. Then what will happen?
Then we can go for tetrad dissection, I guess.
Yeah, that is I knew that will be what you will say. So think of it. In a population where you are inducing the mutation artificially, right? Through, feeding those mutagenic chemicals. So in a population, perhaps, 10 in 100 will be mutated. Okay. And as I discussed about genome coverage, you are likely to screen 1 lakh plus colonies. Right? So then you will have to do tetrad dissection on those 1 lakh. हाथ वहीं पे जमा करके आना पड़ेगा. Tetrad dissection is very, very tedious job. So what is generally done, therefore, all screens begin with haploids, unless you have a specific reason to believe, like my PhD is in protein synthesis and regulation. Okay, so there we knew that the mutations will be lethal, because if there is no protein synthesis, there is no cell, right? So there we did it in diploid.
Thank you, sir.
Sure. Next question.
Sir, I have 2 questions.
Okay.
So one question is, first one is, why is a recessive allele expressed in homozygous condition? Like what I know about recessive gene is that whether it is not expressed or expressed in low quantity. So how is that recessive allele in homozygous condition going to give any phenotype in a particular cell or in a body?
Madam, what you know is wrong from top to bottom.
Okay, sir.
A recessive allele is not about less amount of production. A recessive allele is about reduced function. Like, I'll give you an example. I'm making it up. If the wild type enzyme turns over 500 substrates in substrate molecules in a minute, the recessive allele might be doing the same for 450 molecules.
Okay.
Now, next question.
Sir, I just want.
No. Iman, just hold on.
Yeah.
You were saying
So
something about
next one is from meiosis. where cell undergoes 2 divisions but replicates only once. So what are the factors which are inhibiting that cell to duplicate it once again while it undergoes the meiosis second?
Okay. So you are assuming, okay, I was taught something, थोड़ा याद रखना.
Yes, sir.
That the saying, this is a typical American saying, that if you assume, you make an ass of you and me. Okay? So never assume. You assumed that there is a mechanism to inhibit second round of replication.
Okay, sir.
So there is no such thing. Okay? It's a completely different mechanism altogether that dictates meiosis. Okay? Meiosis, अगर एक चीज बोलो,
Yes, sir.
आपको क्या लगता है आपके स्किन सेल में मायोसिस होता है?
No, sir. Only mitosis.
तो मायोसिस किधर होता है फिर?
Sir, germ cells में.
So they have a very specialized mechanism for that. Okay, and I can. Okay, one thing that I would like to inform you all, please understand that I do not know everything and I am not shy about it. I know only a specific set of things and which I know well. Okay, so meiosis is one such thing that I don't know much about. Okay, and because it is not really a core part of the course, therefore I did not bother to learn it again. How many of you have read Sherlock Holmes, Arthur Conan Doyle, Sherlock Holmes? So you know that Sherlock Holmes did not know that sun rises in the east, and he did not care for that. Okay. So yeah, like I only care for things which I really need to know. The rest are information I can always gather. Acha Imund, bolo.
Sir, I just want to clear doubt on missense and nonsense mutations. Like, what are they? Like, how are we detecting about the different? We know about point mutations. Now, missense and nonsense, like a sequencing and like detecting of target sequence and identification of nucleotide is only like what will be our approach towards missense or nonsense mutants? Sir, if you clarify the doubt.
If you clarify your question, I can clarify my answer. Sorry,
Sir, actually, I'm not very clear about. Sir, actually, I'm not very clear about like missense and nonsense mutations. So, if you can explain a bit about that, and how can we approach to identify the missense and nonsense mutations?
Okay, see, identification of mutation will necessarily involve sequencing, right?
Yes, yes.
Now, okay, fine. If this is something that many of you are having difficulty with, let me get a pen. Just give me a minute. कोल्ड ड्रिंक्स এর মতন লাগছে Okay, I will get in this question if and only if there is no other question about the preliminary, because this is a little bit later chapter topic, but I will be happy to discuss it. As you know that translation I know like the back of my hand, But I will go into it if you do not have any other question about the initial lectures. Is that the case?
Sir, I had some basic questions. You explored about haploid, diploid, aneuploid, and polyploid. So I did not got like a very sound understanding about aneuploid and polyploid. So if you just elaborate a bit.
See, the ploidy is defined by the singular set of genes or chromosomes, singular set of chromosomes. Okay, so if you have only one set, it is haploid. If you have two sets, that is diploid. Artificially, if you have one extra chromosome, then it is polyploid. Okay, it is not that. See, there are organisms like frogs. Okay, I think they have 4 sets of chromosomes. They are 4n, I think. I may be mistaken. Please, Shaura, please put a note to that. I will look it up and send it to you. Okay. The question is whether frog has 4n or not. I may be wrong. I will confirm it. So an aneuploid is that from your whatever you see, you are a diploid animal. Okay. If for some reason one of the chromosomes was missing in your cell, then it will be an aneuploid. It is not that you will have no chromosome. Then there is no cell. Okay. Anything else? Okay. So your next okay. Here are 2 things. Your next discussion starts at 12. I think it is good that you have a break in between. And this codon and this sense nonsense, missense nonsense mutation will take at least an hour. Okay. So I would suggest that we have another separate discussion hour just to discuss that. And I have no problem. I am literally working from home. So if you want 1 in the morning, I am absolutely fine with that. I exchanged WhatsApp messages with some of you at 1 in the morning yesterday or today. So any time of the day is fine for me. Generally, evening is better. Okay. So I would like to emphasize on one thing. So far, my experience of teaching PhDs and MTechs have been very, very good, very rich and rewarding. I enjoy it. But you should know about one thing, that I do have a very high standard. Okay. You are going to earn a PhD. Okay. So you should be literate. Okay. Therefore, I will very thoroughly examine you on your concepts. Since you are working from home, I would request that you please read listen to the videos multiple times. It is so much better, right? You have access to the whole lecture series. Like I have uploaded the PPT also, right? Do you all have the PPT as well? So you know what are the slides that are coming. You know the textbook. I have tried my best to explain it to you. And then we have discussion hours. If you want, I can have more discussion hours. All of that is acceptable. The only thing that is not acceptable is that you expect to pass the course without learning the subject. Okay, that is not acceptable. I will be very tough when it comes to grading. Okay, this is the reason why you have seen, I do not know what is happening in other courses. In my course, I decided that the majority of the grading will be based on viva voce, so that I can ask you and get your answer on the spot. So that is actually even more challenging for you, because in a typical exam, if I pose a complicated problem, you have 20 minutes, 30 minutes to think through and then write the answer. Here in 10 minutes, you will have to actually convince me that you have understood what I taught. Okay, so for that, what is required from your end is hard work. Okay, I would not be shy at all. See, this is a 9-credit course, right? At IIT Kanpur, the credit system is defined as number of hours you are required to give for a particular subject. So in this case, it is 9 credit, meaning you have to devote 9 hours per week. Okay, so I will hold you to that standard. Okay, like right now, as you can see, that any question you ask, I will give you answer. Okay, but based on that, you will have to study on your own, the textbook, the lectures, any other resource that you get on the internet. You bring in all sorts of questions. I will try to answer all of those. Okay, but then I will ask you questions. And the 2 assignments that we will have, I really do not care, do not mind with how many people you discuss. You can discuss with anybody and everybody, because by discussion also you will learn. Okay, and my assignment questions will never be that you will be able to copy from someone and or you will find the answer somewhere. These are all typically designing experiments and interpreting data type questions. So you will not find it anywhere. You will be able to discuss with people who will teach you how to do that, who may be able to help you in thinking about. And I will, of course, teach you along the way before I give you such assignments. But it is not at all a bad thing that if you, in your effort to solve the assignment problems, you take help from someone else. That is absolutely all right. But you should write in your own language. Okay, whatever you do, write in your own language. So you tell me that when should we do this other discussion hour? So tomorrow you have one discussion hour at 8 to 9 in the morning, and then do you have any other discussion hour tomorrow? Okay, then let us schedule one from 11 to 12 tomorrow. How is that?
Yes.
Is it fine?
Fine.
Yes.
Yes, sir.
So 11 to 12 tomorrow, we will have the other discussion hour. Okay. And Shora, you please circulate the note to the whole class. Like,
Okay, sir.
you can also
Okay,
send email to the same thing, bsc651a@iitk.ac.in. Because I want all of you to get answers to those questions. Okay, I will not give you the answers. You try. If you can't find the answers, then I will help you.
Okay, sir.
Okay? And in this format, this is for the entire class. In this format, it is very easy for you to stay under the radar. That is, not asking any questions or not interacting. It is very easy. But please understand that these courses are for your benefit. Therefore, actively participate actively learn, and you will enjoy it. Okay. Once you understand the concepts of cell and molecular biology, you will feel empowered. Let me put it that way. Like when this entire world was fighting COVID, this is the one skill set that people were heavily dependent upon, right? Understanding genetics and understanding cell biology. So this is your opportunity to learn it well. So do not, waste that opportunity. Okay, if you can make me work very hard, I'll be very happy. Okay, chalo, bye. So we'll see you tomorrow at 11 o'clock.
Sure.
Okay.
Okay, sir.
Yes, sir.
Thank you.